Good afternoon, all of you. I welcome you all to the Model School webinar Junior Intermediate channel. We have been running these sessions since very long. Today we covered almost 190 sessions for different subjects by now. As part of this webinar program, we are running botany classes also. So I welcome all the botany lecturers, PGTs, BPC first year students. Today we are having Srimati L.BHARATHI, PGT TSMS Jarasangam. She is very efficient and very obedient, sincere to the work to the core. She taught for both first year and second year. As and when we need the services of the PGT, she is the first one to raise her hand. We are very thankful to her. We are in the sexual reproduction of the flowering plants chapter. Reproduction is very important for all the living organisms. Reproduction only leads to the progeny. Without progeny, there is no life. As part of that, earlier she discussed about the male reproductive organs in the flowering plants. Today she will discuss about the female reproductive organs in the flowering plants. In further classes, she will discuss about the pollination, fertilization, pre-fertilization changes, post-fertilization changes, etc. So it gives me immense pleasure to invite Bharati to present this session. At this juncture, I would like to thank Srimati BDRS Manjari, who is hosting these sessions without any break. We are very much thankful to her. So I welcome Srimati Bharati.
Thank you, sir.
Start, Jayama.
Okay, sir. Thank you, sir. I thank my principal, Mohammad Riazuddin, TSMS Jarasangam. I thank my subject coordinator, A. Venugopala Reddy, sir. I thank the dignitaries behind the screen who has given me this opportunity to deliver the lecture on the chapter 7, Sexual Reproduction in Flowering Plants, under the unit 3, Reproduction in Plants. So in the previous class, we have learned about the pre-fertilization structure. In the pre-fertilization structure, we have learned about the stamen, microsporangium, and the pollen grain. In today's topic, we'll learn about another structure, that is female structure in the pre-fertilization step. So that is the pistil, megasporangium, and the embryo sac. So pre-fertilization structure and events is a part of the sexual reproduction that is taking place in the plants. So we know that with the help of sexual reproduction only, there is a variation in the progeny. In earlier classes and lower classes, also in the 10th class, you have learned about the structure of flower. So in the structure of flower, you have learned about the 4 whorls that is present in the flower. The flower is generally developed on the swollen part of pedicel that is called as thalamus. Thalamus, it is also called as torus or the receptacle. So on the thalamus, the floral structures are present. This flower is considered to be the modified shoot. In the modified shoot region, the florigen, if it is available, then only flower primordium will develop and it will bloom into the flower. So the flower consists of 4 whorls. Outermost whorl is a sepal and next to sepal is a petal. Sepal is generally green in color and the petal is brightly colored. So the petal useful nature is that it is attractive, attracts all the living organisms alike. For example, if you see the insects which comes and visits the flower is because of the attractive nature of the petal. So sepals and petals are present in the outer whorls of the flower. Now these sepals and petals, even if they are not present, the plant can survive, like in grasses. If you see in the hibiscus flower, the petals and sepals are present. But in few plants, even if the petals and sepals are absent, then also sexual reproduction will take place. How does the sexual reproduction takes place in them? It is because of the presence of andrecium and gynecium. So andrecium, in previous class you have learned about the stamen. Stamen consists of two parts, that is anther and the filament. You have learned about this male reproductive structure, and inner to this male reproductive structure is the female reproductive structure, that is called as gynecium. The other name for gynecium is nothing but the pistil or the carpel. So pistil or the carpel is nothing but the female reproductive structure of the flower. It is very essential part of the flower through which the seeds are formed. So, you know, if we sow the seeds in the soil, then only the seedling will develop and the plant is formed. So gynecium has to be protected more than other structures. So gynecium is generally present in the center of the flower, surrounded by these other three whorls. So gynecium is very important for the formation of the progenies, that is nothing but the zygote. So the gynecium consists of three parts. It is a stigma, style, ovary. So pistil is also known as gynecium. So the pistil contains three parts: stigma, style, ovary. In your 10th class, you have learned about the structure of pistil. So you may able to recollect by looking at the diagram that stigma is present at the tip. In the stigma only sticky substance is present, which will help in attracting the pollen grain. So it is showing chemotactic, that is chemotropism. In 10th class, you have learnt about the tropic movement and the nastic movement. So stigma is showing the chemotropism. What is chemotropism? Chemotropism means the stigma is releasing the chemicals by which the pollen grain will be attracted towards it and it will develop the pollen tube. Next part of the pistil is the style. Style, you know, it is an elongated portion, and generally the style will decide whether the pollen tube should grow through it or not. Where is the pollen?
ఎగ్జిట్ అయినట్టుంది కదా,
తనకేదో డిస్టర్బెన్స్ వచ్చింది.
జాయిన్ అవుతుంది. భారతి మేడం,
Stigma is the landing platform of the pollen, and style is an elongated slender part, and the ovary is a basal bulge part which will have the ovules. So if you see the diagram again, the stigma is a landing platform for the pollen. On this stigma only the pollen grain will fall. So when it falls, it will enlarge the pollen tube. And style
భారతి
is
మేడం స్క్రీన్ షేర్ చేయండి. స్క్రీన్ షేర్ ఆగిపోయినట్టుంది. అయ్యో,
if you see this structure, the ovules are present inside this swollen and bulge part of the ovary. So the pistil contains three parts: stigma, style, ovary. And the andrecium contains two parts: anther and filament. Now we'll exclusively learn about the pistil, megasporangium, that is ovule, and the embryo sac. So going details into the details of the gynecium. So I told you pistil is nothing but the gynecium only. Only if one pistil is present, it is called as monocarpellary. Carpel is nothing but the gynecium. Only if one is present, it is called as monocarpellary. Example for this is hibiscus, that is nothing but the shoe flower, and the papaver somniferum. So papaver somniferum and the hibiscus will have only single pistil. And if more pistils are present, it is called as multicarpellary. More pistils, it is called as multicarpellary. So example for this, annona and mycelia. So if many pistils are present, they may be free, they may be fused. So if the pistils are fused together, they are present, it is called as syncarpus. Syncarpus. So fused pistil is called as syncarpus. Example for this is papaver. And free pistil is called as apocarpus. Example is mycelia. The another example for syncarpus is datura also. And another example for apocarpus is annona. So single pistil, monocarpellary, example hibiscus, papaver. More pistils, multicarpellary, annona, mycelia. Fused pistil, syncarpus, example papaver. Free pistil, apocarpus, example mycelia. So see that pictures now you can easily understand. So hibiscus is the shoe flower or the hibiscus. You know mandara poo, is it not? That flower only, hibiscus flower. So in this you can see single pistil. This is a single pistil encircled by the anthers. Andrecium. This is the anther, this stalk is the filament. So in the hibiscus flower in the center, you can see the single pistil. And mycelia, this is a mycelia flower. And in the mycelia, there are many pistils. Each one is a pistil. This pistil is fused. Okay. The pistil in the mycelia is not fused, it is free. Okay, this is the papaver flower. Papaver somniferum. In this, the pistil you can see in the center, it is fused. This is a fused pistil, that is syncarpus in the papaver. So papaver with the fused pistil is present. It is called as syncarpus. Mycelia, if you see in the center, the carpels are free. It is called as apocarpus. Free pistil or apocarpus. Example for syncarpus, papaver. Example for apocarpus, mycelia. Now, you can see there is a cavity inside the ovary. So that ovarian cavity is called as locule. You can know locule or the ovarian cavity is present inside the ovary. So if only one locule is present or one cavity is present, it is called as unilocular, like in beans. You have seen the beans fruit, no? Only one locule will be present. If you cut open the tomato, it may contain two locules. It is bilocular, like that. So ovarian cavity contains locule. The locule will have the placenta in it. So inside the locule, what is present? Inside the locule, placenta is present. What is placenta? Placenta is nothing but a mass of cells which will join the ovule with the ovary wall. So placenta is helping the ovules to be connected with the ovary. That is called as placenta. So from the placenta, what will arise? The megasporangium or the ovules. Megasporangium is nothing but the ovules. It is not the ovary. Megasporangium is the ovules. So there may be single ovule or many ovule. Single ovule means in the locule, only one ovule will be present. Example for this is wheat, paddy, sunflower. So generally in these examples, only one locule will be present, that is it is unilocular, and single ovule will be present. The example for this is wheat, paddy or sunflower. Sometimes many locules will be present either in the one locule or multiple locules. So example for many ovules, is called as papaya, watermelon, orchids. So if one ovule is present, generally one seed will be present. If many ovule is present, many seeds are present. So in papaya fruit, if you cut open, there will be many seeds, no? That means many ovules were present before the fertilization has taken place. So like that, how many ovules are there? That many seeds may form in the fruit. So single ovule is seen in wheat, paddy, and the sunflower. Many ovules is seen in papaya, watermelon, and the orchids. So if you see this structure, it is the TS of ovary. Inside the ovary, you can see the bulged portion. This bulged portion is nothing but the ovules. These ovules are connected to the ovary wall by a cushion-like parenchymatous structure that is called as placenta. So inside the ovarian cavity, you can see the space. This is the space. So it is locule 1, locule 2, locule 3. That means it is having 3 locules. So in 3 locules means trilocular ovary. So inside the locule, what is present? Ovules are present. How many ovule is present in each locule? It is 2 ovules. It is connected to the ovary wall by this placenta. Likewise, it may have only single carpel, single locule may be present with many ovules, or it may have many locules with many ovules. So it can be unilocular like this or trilocular like this. So locule is nothing but the ovarian cavity inside with the ovules will be present attached to the ovary wall by the placenta. And this is the rice grain. This is nothing but the example for single ovule. So from the single ovule, single seed will arise. So it is example for this is rice. And you can see here many seeds are present. Many seeds means many ovules were present before the fertilization process. So many ovules like many seeds as in papaya fruit. So you know papaya fruit. You eat, it is good for the eyes. Vitamin A is rich in it. So you can see if you cut open papaya fruit, there will be many seeds. That means many ovules were present before the fertilization process. Now we'll study about the structure of ovule. I told you ovule is nothing but the megasporangium. What is microsporangium? It is present in the pollen grain, is it not? So pollen grain is present inside the microspore, is it not? So megasporangium means ovule. So megasporangium contains integument. That is why it is called as integumented megasporangium, nothing but ovule. So what is ovule? Ovule is an integumented structure present in the ovary. Where is ovary present? Ovary present on the thalamus of the flower. So ovary contains ovule. It is called as integumented structure. Why it is called as integumented structure? Because ovule is essential for the formation of the seed. That is why it should be protected. That protecting covering or the layer is called as integuments. That is why it is called as integumented structure. And ovule act as megasporangia. It is called as megasporangia and integumented structure, or in short, integumented megasporangia. So in the exam, if they ask what is the integumented megasporangia in the flower, means it is a ovule. So what is the connection between the ovule and the ovary? It is through the parenchymatous tissue called as placenta. So placenta will help in the connection of the ovule with the ovary. It is joined by the placenta by the stalk called as funiculus. The ovule is attached to the placenta of the ovary by a stalk called as funiculus or the funicle. So the stalk of ovule, what it is called as funiculus. Like stalk of flower is called as pedicel. Stalk of inflorescence is called as peduncle. Like that stalk of ovule, it is called as funiculus. So The point of attachment of the ovule body with the funiculus is called as hilum. So if you see the ovule, there are two parts. One is the stalk, one is the body. So stalk is called as funiculus, and the area where the body of the ovule is attached to the funiculus is called as hilum. What is hilum? The body is attached to the funiculus by a region. That region is called as hilum. It is nothing but the junction of the ovule and the funiculus. So ovule and funiculus, in between them will be present the hilum. And what is present between the ovule and the ovary? That is nothing but the placenta. And this placenta is attached to the ovule by a stalk called as funiculus. Okay. So ovule will also have one or two protecting envelopes, that is wall layers. It is called as integuments. These integuments can be single, can be double. But in few ovules, there are no integuments at all. Like in gymnosperm. In gymnosperm, only one integument is present. Without the integument, the ovules may be present, like in parasitic plants. So ovule may have one or two protective envelope. It is called as integuments. These integuments are absent at a certain point. That tip is called as micropyle. So this is very important. What is micropyle? Micropyle is a region where the integuments are absent, or it is just a pore of the ovule. So this is the structure of ovule. If you see this structure of ovule, this whole thing is the body of the ovule, and this is the stalk of the ovule. The stalk of the ovule is called as funiculus. And this is the body of the ovule. This body of the ovule is attached to the funiculus in a region. This region is called as hilum. Okay. So this hilum is extended into the body of ovule. It is called as raphe. Funiculus extends into the body of ovule. It is called as raphe. Okay, along the ovule body it is present. It is called as raphe. And this funiculus and the raphe will have the vascular strand for the supply of food and water minerals. And if you see the body of ovule, in the body of ovule, integuments are present. These are the integuments. This is inner integument. This is the outer integument. If you see, two integuments are present in this photo. So at the base of the ovule, this is the base of the ovule opposite to the micropyle. What is micropyle? Micropyle is the opening of the ovule. Here there are no integuments. So the micropyle opposite end is called as chalaza. Chalaza is nothing but the basal part of the ovule. Micropyle and chalaza are always opposite to each other. This side is called as chalazal end and this side is called as micropylar end. So in the micropylar region, you will find the embryo sac. This is the embryo sac. So we'll learn about the embryo sac later on. Now we'll recollect what we have learned till now. So opposite to the micropylar end, at the base of the ovule is the chalaza. So what is chalaza? Chalaza is nothing but the base of the ovule. And inside the integument, it has a mass of cells. This mass of cells is called as nucellus. See the spelling? It is not nucleus. It is nucellus. What is nucellus? Inside the integument, that is the mass of the ovule is made up of nucellus tissue only. Now, inside this nucellus tissue only, embryo sac will be present. We'll learn about that now. So, on the basis of integument, ovules are three types. What are they? Unitegmic ovule, bitegmic ovule, ategmic ovule. Unitegmic ovule is nothing but it has only one integument. Example for this is Helianthus or Datura. So Helianthus means sunflower. It belongs to Asteraceae family. or the Compositaceae family. Datura belongs to Solanaceae family. So in Solanaceae family and Compositaceae family, what is the nature of integument in ovule means what will you write? It is unitegmic ovule. It has only one integument. Bitegmic ovule means two integuments are present. So outer integument is present, inner integument is present. So example for that is monocot and polypetale members. So monocot means you can take rice, wheat like that. Polypetale members means you can take the hibiscus, that is the shoe flower. So in them, how many integuments are present? They have two integuments, outer and inner. It is called as bitegmic ovule. In the unitegmic ovule, the gymnosperms are also present. So ategmic ovule means they don't have any integument. So it is seen generally in the parasitic plants. That is, example for this is Loranthus santalum also, that is sandalwood also. So the integuments may be present, may be absent. The nature of the integument can be one layer or two layers or completely absent. What is the function of integument? It will protect the growing embryo sac present in the nucleus tissue. The nucleus will have nutrients for the embryo sac or the female gametophyte. Female gametophyte means it bears a female gamete. What is the female gamete? It is nothing but the egg. So, female gametophyte is nothing but the embryo sac. Inside the embryo sac, it bears the female gamete or the egg. So, while seeing the structure of embryo sac, we will study about the egg also. So this picture you can see bitegmic ovule. That means it has two integuments. Outer one, that is the blue color, is the outer integument. The green color is the inner integument. This is the ovule mass of the tissue, that is nucleus. So it is having two integuments. Example for this is monocot or the polypetale members. In this, if you see, only one integument is present. It is called as unitegmic ovule. So example for this is present in gymnosperm, then Compositaceae family and also Solanaceae family. They are shown in two different colors. So it is having only single integument. If you see this bifid, that means both the integuments are covered by again another layer. It is called as bifid. This is not there in your syllabus, so you can avoid it. These are the example. Datura, it contains unitegmic ovule and Datura belongs to the family Solanaceae. So in Solanaceae, the ovule will have only single integument. This is the picture of sunflower. It also has single integument. It is called as unitegmic ovule. This is the bitegmic ovule in the rice plant. Rice plant, it is nothing but the monocot. So in monocot, bitegmic ovule is seen, that is it has two integuments, both outer and inner. Why do you think it has two integuments? Because it has only single ovule inside. That means only single seed is formed. That should be protected. That is why it is covered by two layers for protection. This is ategmic ovule as seen in the Loranthus. These flowers, if you see, it is nothing but the Loranthus flower only, the plant flowers only. So, ategmic ovule is seen in Loranthus or Santalaceae. That is nothing but it is a parasitic plant. Now we'll see various types of ovules. Depending upon the orientation of the body of the ovule, you can find various types of ovules. Generally, there are 6, but in your syllabus, they have given only 3. So we'll go with it. So orthotropous ovule, anatropous ovule, campylotropous ovule. What is orthotropous ovule? Orthotropous ovule is also called as atropous ovule. In this, it is called as straight ovule. Straight ovule depending upon the position of micropyle, chalaza, funiculus. So, when the micropyle, chalaza and the funiculus are in a straight line, they are called as orthotropous ovule. So, In the orthotropous ovule, it is the oldest and the most primitive ovule. It is seen in polygonum. So in gymnosperm also, orthotropous ovule is present. So straight ovule is nothing but atropous ovule or the orthotropous ovule. What is the condition in this? The micropyle, chalaza and the funiculus are in straight line. If you come to the second ovule, it is anatropous ovule. Anatropous ovule means it is completely inverted. That means the micropyle will be lying close to the funiculus. That means it has inverted 180 degrees centigrade. So example for this is sunflower, helianthus. So anatropous ovule means other name is inverted ovule. Generally 80% of the angiospermic flowers will have anatropous ovules only. Campylotropous ovule, in this the embryo sac is slightly curved. The body of the ovule is right angle to the funiculus. Example for this is the beans. In this, the embryo sac itself will be curved. Okay. So micropyle will lie closely to the funiculus, but not as close like the anatropous ovule. So campylotropous ovule is seen in beans. These are the 6 types of ovule. In your syllabus, you have this atropous ovule, anatropous ovule, and campylotropous ovule. So atropous ovule is also called as orthotropous ovule. So in this, the micropyle, the chalaza, and the funiculus are in the straight line. That is why it is called as orthotropous ovule. If you see this anatropous ovule or the inverted ovule, that is, it is having the micropyle lying close to the funiculus. And this micropyle and the chalaza are in the straight line, but not with the funiculus. And if you see this campylotropous, the embryo sac is slightly curved. Okay, that is why it is called as campylotropous. So example for atropous is polygonum, anatropous example is sunflower, and campylotropous ovule is beans. Megasporogenesis. So what do you mean by megasporogenesis? Earlier we have learned about the microsporogenesis. So microsporogenesis means production of microspores from the microspore mother cell. That is called as microsporogenesis. So now you can guess what is megasporogenesis. Megaspore developing from the megaspore mother cell is called as megasporogenesis. Now megaspore tetrad is formed from the megaspore mother cell by which division? By the meiosis. Meiosis is also known as reduction division. So megasporogenesis is nothing but the formation of megaspore from the megaspore mother cell. There we have seen microspore mother cell, also known as pollen mother cell. In there we find the microspores. Here we find the megaspores. What will be the future nature of the megaspore? It will form the embryo sac. So that also we will learn now. And nature of megaspore mother cell is that it is a large cell containing dense cytoplasm and the prominent nucleus. It has lot of cytoplasm with granules and a prominent nucleus because now it has to form the megaspores. Now, megaspore mother cell is formed from a single cell in the nucleus. So, from the nucleus tissue, one cell near the hypodermis, that is near the micropyle, will enlarge to form the megaspore mother cell. This megaspore mother cell will later on form the linear megaspore tetrad. That means in a line they are formed. So we'll learn about that now. First, we'll see the answer for this question. What is the importance of megaspore mother cell, which is undergoing meiosis? So when megaspore mother cell is present, it is nothing but part of nucleus tissue only. Nucleus is diploid and megaspore mother cell is also diploid. So when this megaspore mother cell is undergoing meiosis, what will happen? We will get 4 haploid megaspores. So when a meiosis is occurring, we know 4 daughter cells are formed. So it also forms 4 megaspores. What is the nature of megaspore? Because it is undergoing meiosis, reduction division. That means it is haploid. From diploid megaspore mother cell, we'll get haploid megaspores. So ploidy of megaspore mother cell is 2n, that is diploid. And ploidy of megaspores is haploid. How many haploid megaspores are formed? 4. Okay. So we'll see megasporogenesis. Megasporogenesis is nothing but the formation of megaspore from the megaspore mother cell. Where is this megaspore mother cell present? It is present inside the nucleus tissue. Okay, in the body of the ovule, nucleus tissue is present enclosed by the integuments. So one cell near the micropyle will enlarge and form the megaspore mother cell or the hypodermal cell. That cell will enlarge, it will have a dense cytoplasm and a prominent nuclei. And this 2n condition will undergo meiotic division and form 4 haploid megaspore. From the 4 haploid megaspore, 3 will degenerate. That means it will dissolve, it will disintegrate. Only one functional megaspore will be present. What is the ploidy of megaspore now here? It is N. That means haploid. That means it has only single set of chromosome. So after formation of megaspore, it will help in the formation of one embryo sac. So megaspore will undergo mitosis and form embryo sac. The ploidy of this embryo sac is nothing but haploid. Inside this embryo sac, you will find the egg. So what is egg? Here it is a female gamete. So there in the pollen grain, we have seen male gametes are present. Inside the pollen grain, male gamete are present. What is pollen grain? Pollen grain is a male gametophyte. Here embryo sac is a female gametophyte. So inside this embryo sac, egg is present. So egg is the female gamete. Embryo sac is the female gametophyte. So formation of megaspore from the megaspore mother cell is called as monosporic development. What is monosporic development? If the megaspore is formed from the megaspore mother cell, single megaspore mother cell, it is called as monosporic development. So embryo sac, the embryo sac is also known as female gametophyte. What is embryo sac? Embryo sac is nothing but it carries the embryo. Where is the embryo formed? Inside this sac-like structure of the ovule. So inside the ovule, embryo sac is present. Inside the sac, the egg is present. After the fertilization of the egg, the zygote is formed and zygote will develop into embryo. So functional megaspore forms embryo sac or the female gametophyte. So method of development of embryo sac from a single megaspore is called as monosporic development. Monosporic development means from a single megaspore, embryo sac will develop. There are also tetrasporic development also. So generally it is a monosporic development seen in the flowering plants. Mono means single. Sporic means spore. What is the spore here? Megaspore. Single megaspore will help in the development of embryo sac. That is called as monosporic development. So what is the ploidy of the cells of the nucleus? Megaspore mother cell, functional megaspore, and the female gametophyte. So nucleus is the tissue present in the ovule encircled by the integuments. So nucleus is the diploid. It is 2n. And from the nucleus only the hypodermal cell will form the megaspore mother cell. So nucleus is 2n means megaspore mother cell will also be 2n. So when this megaspore mother cell will undergo meiosis to form 4 megaspores, out of this, 1 megaspore is functional. It is having the ploidy and haploid because from megaspore mother cell, reduction division or the meiosis has occurred. So 4 megaspores are formed. All the 4 are N. So functional megaspore is also N, that is haploid. Inside this functional megaspore, female gametophyte or the embryo sac is present. It is also N. Okay. Inside the female gametophyte, egg is present. Egg is also haploid. So all the gametes will be haploid only. So we'll see how is the embryo sac formed now from the megaspore. So the nucleus of the functional megaspore will divide mitotically. Mitotically, by mitosis, the nucleus will divide to form two nucleate embryo sac. So from megaspore, two nucleate embryo sac is formed. Now there is no cell wall deposition around this nucleus. Again, this two nucleate structure will divide mitotically to form 4 nucleus and later on 8 nucleus of the embryo sac. So inside the embryo sac, now how many nucleus are present? 8. So it is having 8 nucleus without any wall formation. Cell walls are not formed now. So these are called as free nuclear mitotic division. That means the formation of the nucleus from the megaspore to 2, then 4, then 8 is called as free nuclear mitotic division. After this stage, after formation of 8 nucleus, the cell walls will be laid around 6 nucleus and remaining 2 nucleus will not have any cell wall. So it is called as 8 nucleate 7 celled embryo sac. So we'll see that now. So after this free nuclear division, mitotic division is over, cell wall is formed around this nucleus and then it forms a typical female gametophyte or the embryo sac. So female gametophyte or the embryo sac is ready for us now. So inside this female gametophyte or the embryo sac, how many nucleus are there? It is having 8 nucleus. So out of this 8 nucleus, 6 nucleate are surrounded by cell wall and the remaining 2 nuclei which does not have any cell wall is called as polar nuclei. Okay children, I'll repeat once again. So out of the 8 nucleus, 6 will have the cell wall and remaining 2 will not have the cell wall formation. It is called as polar nuclei. And this below the egg apparatus, these 2 polar nuclei are present in the large central cell. So the central cell is present in the center of the embryo sac and that central cell will have these 2 polar nuclei. So we'll see the structure, that time you can understand better. So now we'll see the formation of megaspore. So megaspore formation is from the megaspore mother cell is called as megasporogenesis. So this is the nucleus tissue. In the nucleus tissue, one cell near the micropyle will form the hypodermal cell or the megaspore mother cell. Inside it, dense cytoplasm with a prominent nucleus present. So this forms the megaspore mother cell. This megaspore mother cell will undergo meiotic division to form tetrad, linear megaspores. These are the 4 megaspores. Now these 4 megaspores are haploid now and they are present in a linear manner. Out of this 4, 3 will degenerate. Only 1 will be functional. This functional megaspore is a haploid now. This in future will form the embryo sac. From this, if the embryo sac is formed, it is called as monosporic development. So we have got the functional megaspore. Now this functional megaspore will undergo division, mitotic division. It will first form 2 nucleus. Here it is not shown, but from single megaspore, from this megaspore, this nuclei will divide mitotically to form 2 nucleate structure. So first 2 nucleate megaspore is formed, then later on it will divide again mitotically to form 4 nucleate stage of the megaspore. So now how many nucleate are present? 4. Okay. That was mitotic division only. Again, mitotic division will occur in this nucleus and 8 nucleus will be forming in the megaspore. So now you can count also 1, 2, 3, 4, 5, 6, 7, 8. you can see 8 nucleus is present. Now, this 8 nucleus will not be having the cell wall till now. But as it is maturing, it will be laid down by the cell wall. So you can see cell wall is present around this structure, and later on this structure will also develop the cell wall. So if you see here, there are 8 nucleus, 1, 2, 3, 4, 5, 6, 7, 8. What will happen means, after the formation of 8 nucleate stage, one from the upper side and one from the lower side will move to the central part of the megaspore. After movement of the 2 nuclei to the central part, cell wall formation will be taking place. And then at the top and near the micropylar end, I told you near the micropylar end, megaspore mother cell will undergo reduction division. So at that end, the 3 nuclei will develop cell wall and form 2 synergids and 1 egg. This structure is called as egg apparatus. Why it is called as egg apparatus? Because it is having the egg. Apart from egg, it is also having the synergids. So this is at the micropylar end. Now if you see the next diagram, micropylar end has been shown at the bottom. So don't get confused. Micropylar end is having the egg. So wherever micropylar end is present, there only the egg will be present. So at the micropylar end, 3 cells are formed. At the lower chalazal end, also 3 nuclei are present, which will develop cell wall layer. And then 3 nuclei will be surrounded by the cell wall to form antipodals. So here the chalazal end, you can see it is having 3 antipodals. Okay, children. So at the micropylar end, you will have the egg apparatus. At the chalazal end, you will have the antipodals. So this central part is called as central cell. Inside the central cell, you have the free migration of the nucleus. It is called as polar nuclei. So central cell will have what? 2 polar nuclei. Okay, children. Now, how many nuclei are present? 1, 2, 3, 4, 5, 6, 7, 8. 8 nuclei are present. Now, how many cells are present? You can count 1, 2, 3, 4, 5, 6, and this large cell 7. And this is called as embryo sac. So this embryo sac is nothing but female gametophyte. So this female gametophyte is nothing but the embryo sac. It is the mature embryo sac which will have the egg now. Okay, children. So embryo sac will have 8 nucleus, 7 cell embryo sac. That is, you have to remember, it is very important. So again, I will repeat, it is 8 nucleate, 7 cell embryo sac. Okay, we'll learn more about this again. So we'll have a recollection of what we have learned till now. Structure of embryo sac. It has 3 cells at the micropylar end. It is called as egg apparatus. 1 egg cell, 2 synergids. The synergids are nothing but it is having filiform apparatus which will guide the pollen tubes towards the egg. So if you see synergids, these synergids are 2 in number and they are present laterally to the egg. What is the nature of this synergid? It will have the nucleus towards the micropylar end and the vacuole towards the central cell. The synergids will have nucleus towards the micropylar end and the vacuole towards the central cell or the chalazal end. And this synergids will have a finger-like projections into it. It is called as filiform apparatus. What is the function of filiform apparatus? Means it will help in the direction or the movement of the pollen tube towards the egg. So when the pollen tube is approaching, this filiform apparatus will guide the pollen tube inside the embryo sac. So when this pollen tube has reached the tip, then one of the synergids will disintegrate. Okay, so this disintegration will help in the opening of the cavity. Then the pollen tube will release its male gametes. So synergids will help in a chemotactic manner to attract the pollen tube, and then when the pollen tube is entering, one of the synergid will disintegrate so that the pollen tube can easily enter into this embryo sac. So next is the egg. So what is the egg? You know, it is a female gamete. Female gamete is nothing but the egg. Where it is present? Egg is present inside the embryo sac. What is embryo sac? Embryo sac is a female gametophyte. Okay. So what is the function of egg? It helps in the process of zygote formation. That is the function of egg. What is the nature of egg? Nature of egg, the nuclei present towards the central cell and the vacuole is present towards the micropylar end. Nuclei will face towards the polar nuclei or the central cell, and the vacuole will be towards the micropylar end. So both of them, the synergids and the egg, will have polarized protoplasm. Next is the central cell. If you see the central cell, it is large, present in the center, enclosing two nucleus. This two nucleus is called as polar nucleus. So at the in the center, there is a large central cell with two polar nuclei. So two polar nuclei is present in the center. So what is the function of these polar nuclei? Means when two male gametes, there are two male gametes in the pollen tube. So one male gamete will fuse with the egg. What is the other male gamete do? The other male gamete will fuse with this polar nuclei. So before that, the polar nuclei will fuse with each other to form secondary nuclei. This is N, this is N. Polar nuclei both are present. It will fuse together to form secondary nuclei. So later on, by mitotic division, this central cell, the tissue will divide mitotically and nourishes the embryo. Okay. So central nuclei will have now secondary nuclei. The secondary nuclei is ready to get fused with the male gamete. Okay. So next is the antipodal. Antipodal is present at the chalazal end. Generally, function of the antipodal is not known, but they say that the antipodal cells that are present at the chalazal end will show us the extension of the chalaza at the chalazal end. How much region is chalaza present in the embryo, that will be shown by these antipodal cells. And also, these antipodal cells will supply nutrients and the growth hormones to the developing embryo. So that is the function of antipodal cells that has been known till now. So antipodal cells will contain nutrients like carbohydrates, lipids, proteins, oxidases also, ascorbic also, like that. Nutrients are present in the antipodal. So this structure is nothing but the gynecium with the embryo sac in the ovule. So generally you have seen gynecium will contain three parts, that is stigma, style, ovary. You know, this is the stigma, this is style, this is the ovary. Inside the ovary, what is present? Inside the ovary, ovule is present. So this is the integument which you see here, it is the ovule. So inside the ovule, you can see the embryo sac. Okay. Now this has been enlarged in this way. So at one end, it is a chalaza. At the other end, it is a micropylar end. So micropylar end will have the micropyle. It is the opening where the integuments are absent in the ovule. So at the micropylar end, egg apparatus are present. This egg apparatus will contain laterally placed synergids and centrally placed egg in the egg apparatus. And these two are the polar cells, polar nuclei. This polar nuclei will fuse together to form the secondary nuclei. At the chalazal end, you can see the antipodals. Three antipodals are present. So these three antipodals are nothing but the present at the chalazal end. The egg apparatus is present at the micropylar end. What is the nature of this embryo sac? What is a ploidy means? It is N only. Embryo sac is N. It is also called as female gametophyte. So the nature of the embryo sac is 8 nucleate 7-celled embryo sac. 8 nucleate, 7-celled embryo sac. So let us recall what we have learned till now in embryo sac. So embryo sac will have egg apparatus towards the micropylar end and the polar nuclei in the central cell and the antipodal near the chalazal end. So this is a 8 nucleate 7-cell embryo sac. So filiform apparatus, where it is present in the synergids. Now we'll have a review of male and female organs in the flower. So male and female organs, this is a flower. This is a flower. In the flower, the outermost wall is a sepal. Next to inner to the sepal is the petal. Inner to petal is the andrecium. Inner to andrecium is the gynoecium. So in the andrecium is a male organ of the flower and the gynoecium is a female organ of the flower. So the andrecium will have two structures, that is anther and the filament. And inside the anther, there is a microsporangia. Inside the microsporangia, there are the pollen grains or the pollen. Inside the pollen, there is a vegetative cell and the generative cell. So generative cell will divide to form male gametes, that is 2 male gametes are formed. Okay, this is about the male organ. Now in the female, if you see, it is centrally placed on the thalamus of the flower. The female organ will have 3 parts: stigma, style and the ovary. The stigma, style and the ovary is present in the gynoecium or the pistil or the carpel. So ovary will enclose ovule. Inside the ovule, embryo sac is present. Embryo sac is a female gametophyte. So female gametophyte it is called because it carries the female gamete. What is a female gamete? Female gamete is nothing but the egg. So inside the embryo sac, egg is present. Apart from the egg, polar nuclei which fuses to form secondary nuclei is also present for the fertilization. So the male gametes will fertilize with the secondary nuclei and egg separately. So we'll learn that in the double fertilization. So this is just an overview of what we have learned in previous class and today's class. Now it is time for some mind work. So we'll see first question. Name the component cells of the egg apparatus in an embryo sac. So egg apparatus that is present in an embryo sac will have 3 cells. What are they? 2 synergids and 1 egg. What is the nature? They are haploid. Whatever cells in the embryo sac is present, they are all haploid in nature. So egg apparatus contains 2 synergids and 1 egg. Name the part of gynoecium that determines the compatibility nature of the pollen grain. So the pollen grain will fall on the stigma, and from the stigma, the pollen tube will enter through the style. So the compatibility of the pollen grain is decided by the stigma and the style. Draw the diagram of mature embryo sac and show its 8 nucleate 7-cell nature. So in the mature embryo sac, 8 nucleus is present and 7-cell nature is seen. Show the following parts. Antipodal is present near the chalazal end. Synergid is present near the micropylar end. Then egg is present near the micropylar end. Central cell is present in the center. Polar nuclei is present inside the central cell. Name the parts of angiosperm flower in which development of female gametophyte take place. So what are the parts of the flower where female gametophyte is present? It is nothing but the ovule. Inside the ovule, the embryo sac is present. Inside the embryo sac, the egg is present. So where is this ovule present? In the ovary. What is meant by monosporic development of female gametophyte? So monosporic development means the embryo sac or the female gametophyte will develop from the one functional megaspore. What is the nature of megaspore now here? It is haploid. So embryo sac is also haploid. So formation of embryo sac from one megaspore is called as monosporic development. Example for this, you can take it as polygonum. So with this, the pistil, megasporangium and the female gametophyte, that is embryo sac, is completed. So both the sessions which deals with the pre-fertilization structures, that is male structure and female structure, is completed. So thank you children for listening to the lecture. Thank you sir for giving me this opportunity. Thank you one and all. Thank you, sir.